Well, hello again, everybody. This is Ritu Arora. I am at the University of Texas at San Antonio, where I'm leading the research computing department here. And I'll be co-presenting with my collaborator, Suman Sirumala. Dr. Sirumala is a faculty and researcher at the University of Texas at El Paso. And he's in the School of Pharmacy, and he's using COVID-19, he's using BOINC attack for COVID-19 related drug discovery work. And next slide, please. So just to give you all a quick overview of what BOINC attack is, so like our collaborator David Anderson has already spoken about BOINC attack early on, so he made my job easy. So BOINC attack is a BOINC project that routes high throughput computing jobs from the Stampede 2 and the Lone Star supercomputers at the Texas Advanced Computing Center to the volunteered resources in the community. And as the BOINC attack team, we provide the plumbing for the researchers, like TACC and Exceed researchers, to send their jobs out in the community from the resources that we have here. And we are the ones acting like gatekeepers. We are not the ones who are submitting the actual jobs. So there are vetted TACC and Exceed users who can run their single-threaded or multi-threaded applications through BOINC attack. And over the next slide, I will present what are the applications that we support. Could we go to the next slide? Okay, so just to give you a quick overview of what the overall information flow looks like for BOINC attack. So like I was mentioning, there's a Stampede 2 supercomputer and a Lone Star 5 supercomputer at TACC. And like David was mentioning, people would want to use BOINC attack when they have long waiting times or queue wait times on Stampede 2 and Lone Star 5. And then in that scenario, they run a script on the supercomputer that will help them route the job to the BOINC server. And one requirement for BOINC attack is that the applications are Dockerized or they run as Docker containers. So, and one way for us to achieve homogeneity in the way we run the applications was through Marius Milia's project. He provided the plumbing to facilitate the transfer of the images from BOINC server to the clients. So pretty much like we developed additional code, we extended what was already available in BOINC to add the researcher component and also to support additional features like automated creation of Docker images from the source code and such. So once the images or once the source code is Dockerized or containerized, it's meant to be run inside a virtual box running on the volunteers' devices. And we also extended the BOINC attack computing model to support the virtual machines in the cloud because a lot of our users were having issues with GPU jobs. So we wanted to use GPUs, but then at one point we ran into issue wherein the virtual box does not allow PCI pass-through to use the GPUs from the underlying resources. So just to keep things simple for the users, we just extended BOINC attack to run jobs on a cloud computing system that is equipped with GPUs till we find the workaround for leveraging GPUs that are available through BOINC attack. Next slide, please. So I'm very happy to let you all know we have nearly 10,000 hosts and more than 13,000 users associated with BOINC attack project today. And we have the capability to deliver more than 30 teraflops of computing power just through CPUs. We haven't looked into GPU computing power because that's something we're not able to harness right now. And then the list of supported applications or the applications that we maintain for the community are Autodoc Vina, OpenSeas, which is for earthquake engineering. Autodoc Vina is the drug discovery application that Suman would be talking more about. I mean, he's the researcher who has been running, for whose group we had been supporting Autodoc Vina and other similar applications to run on BOINC attack. And then we have BLAST, VetTool, HTC. These are all computational biology related applications. Then GROMACS, LAMP, SNAMD, which are molecular dynamics applications. And BOTI, again, a computational biology application, and OpenFOAM, which is a finite element application. So in addition to these supported applications, researchers who are vetted at TACC and Exceed are allowed to run any applications that they would like to run. And like I was mentioning early on, the frequency at which the jobs are submitted are really in the control of the researchers who are interested in using BOINC attack. And we are just acting as provisioners or providers of BOINC attack system and acting as gatekeepers, educating people how to leverage BOINC attack for their research and providing support. For example, if they have to prepare the jobs to run on large scale, we would provide assistance for that too. And some of the innovations that we made technically while implementing BOINC attack are distributed Greyfish, which is a distributed file management system. So we ran into a challenge that the virtual machine that's hosting the BOINC server ran low on space. And while we were assisting Suman with his research, we realized like there were several GBs of data that would be produced as a result. So we ran into an issue like how to extend the file system that was associated with BOINC attack without bringing down any of the services. So we innovated a tool called distributed Greyfish that's available from our Git repo. So anybody can use it for provisioning distributed cloud management, distributed file management system on the VMs on-prem or in the cloud. And the other tool that we innovated is Midas, which is for automated generation of Docker images from source code. You can bring in your code written in Python, Bash, R, C, C++, Fortran, Java, and provide the recipe for compiling the code. And the Midas system will automatically generate the Docker images so that that can run with Docker, that can run with BOINC attack. So you'll hear more about these tools and other challenges we run into while operating BOINC attack on April 28th. And the next slide, please. I think it's Suman's turn now to take over and tell us more about his COVID-19 research using BOINC attack. Over to you, Suman. Thank you for driving the slides.
Yeah, thank you, Ritu. So I'm going to talk about the project that we ran on BOINC attack. So here, what we as part of this project, we leveraged this volunteer computing to screen billions of compounds against coronavirus proteins. So we are in the pandemic right now, and like, okay, so this is the story about like, okay, several researchers come together to address the current issue. So I mean, we are in the pandemic, and like right now. And there is an urgent need for the medicines, for the therapeutic options for to treat the COVID-19. So if we just look at the drug discovery process, so the drug discovery process, there is a basic research and like there is a initial drug discovery, preclinical and clinical trial stage, and this whole process and like this whole process will take about usually takes about 10 years and like about $3 billion. So that's a lot of money, and there's a lot of time that is involved in a drug discovery process. So the idea here is like to use computing resources and like a neural algorithm to design drugs and like to identify drugs that so that we can bring the drug molecules sooner to the market. So we can use several computer-aided drug discovery methods for this. So here for this project, like, okay, so we had a very big collaboration at the time of the pandemic. So all the Texas researchers, so we all came together. There are several computational chemists and some medicinal chemists. We all came together and talked about like how we can leverage all the resources that we have and how we can try to find some therapeutics really quick. So there are several people from UT Austin, from Texas A&M, and so Texas Medical Branch, me at UTEP, and Baylor College of Medicine. So we all came together and we started collaboration, try to work collaboratively on this project, and we leveraged all the resources. So we have TACC. So TACC has some good, some great computers, supercomputers, but it's not just the supercomputer. We wanted to take everything faster. BOINC attack was super helpful during this project. So because we were when we were running submitting the calculations on TACC clusters, I mean, there were a lot of people who were running at that time. Okay, there was huge load, and like, okay, our simulations weren't executing right away they were taking like 24 to 48 hours. But when we submitted to BOINC TACC, like, okay, we had the volunteer computers available, and like, okay, they were running much faster, and we are getting the results sooner before they execute on the TACC cluster. So this project, we used all the resources. So the TACC was great, TACC clusters were great, and BOINC TACC was super helpful for this project. So just going into a little bit of science of what's happening here and what we were doing. So we were looking at as you all know, the coronavirus. These have these spike proteins. So we were, so if you see inside, like what you see is the RNA material. And the coronavirus, what it does is it go inside the cell and they will replicate and come outside. And like, okay, so one goes in and like a couple of ones that come out. So within the cell, they replicate. So during this replication, so there are several enzymes, several proteins that are involved in this replication process. So we are targeting these proteins that are involved in the replication process so that the replication will be stopped. So if you look at the different stages of COVID-19. So usually when a patient gets infected, like first stage is the incubation, the second stage is a symptomatic phase where there will be, you'll see the symptoms, and this is a viral replication phase, and then there is an early pulmonary phase and late pulmonary phase. So we're targeting at this phase, at the symptomatic phase. Thank God we have now vaccines, but we still, like, there are people still getting infected, and like not everyone gets vaccinated, and like we don't have enough vaccines distribution at the moment. So even though we have vaccines, the therapeutics is important. So our main target is this viral replication process. So we're trying to find therapeutics targeting proteins that are involved in this viral replication process. So when we are doing these calculations, what we are looking at is, here is a protein. We are trying to find a drug target. So there are so many, there are billions of molecules that can be made, and we can choose any of the compound to test it. But in order to do experiments rationally, like what we want to do is, first we wanted to identify the compounds that can really have a chance. So what we're doing is we're trying to take a drug molecule and trying to see whether it will fit within the binding pocket or not. So if it fits the binding pocket, so usually what you see is like the energy is decreased for the complex. So we're trying to estimate this energy and trying to find that will take to the least possible energy. So the one that gives less, the one that releases more energy, that the idea here is like that molecule will have a better chance. So in terms of like, okay, in terms of concentrations, so what we usually say is like, okay, the molecules, something in the micromolar concentration, if you have IC50 values in the micromolar concentration, then the delta G values is about -8.3, but something that is in nanomolar, that's the delta G values are about -12.4. So here we're looking at if a molecule wants to be approved by the FDA, they want the molecule to be effective in nanomolar concentration. So we're trying to find the molecules that can have this much affinity in the nanomolar concentration. If you look at the delta G or free energy, it's -12.4 kilocalories per mole. So those are the molecules that we're looking. Or even if you find something here, the idea is to go pretty quick to this by using by optimizing its interactions. So what we are doing here is, so we have a known target. So we know like, okay, there is a protein and we have a structure of this protein. And we're trying to see like, okay, what is the important binding pockets, their function. And like, okay, we're trying to stop that function of that protein by binding a drug molecule into that binding pocket. So what we're trying to do is trying to find a molecule that can fit within the binding pocket. And so once we know, like if it fits within the binding pocket, we're trying to estimate its energy, how much energy that can be released when this binds into it. So here, if you look at it, so this is a protein molecule. So what we're doing is like, we're looking at the molecule, we're putting that on the surface of the protein, and there are different binding poses. So like we're looking at each position and what would be its affinity for each position. So that's what we're estimating. So on this protein, on each protein, and like we're trying to find the least energy conformation. So the idea here is like those ones will have a better chance. So if you look at a docking program, this has two components. One is a search algorithm, which I just showed up here, where it searches on the protein different poses that can fit in there, and there is a scoring function that actually scores whether the what would be the affinity of the molecule. So this scoring comes from the contacts that each pose makes to the protein. So by evaluating the contacts, we are trying to estimate the energies. So in this case, what we did was we chose 6 different viral proteins. So these are coronavirus proteins that are involved in the replication process. These are decoded from the RNA of coronavirus. So these have these have an important function in the replication process. So we targeted on the 6 proteins, and we wanted to screen because there are no known compounds. So there are like billion compounds, partial library, and like we screened each these compounds against these proteins. So we used both TACC and BOINC TACC clusters along with BOINC TACC clusters to get these results. So after we screened it, like now we have experimentally evaluated these, and we have already identified several compounds that are highly active towards these proteins and like have a ability to stop the viral replication. And we already have a preclinical data, and like we're moving forward with that right now. So here again, like, okay, so BOINC TACC and TACC clusters were used for the project, and BOINC TACC was really helpful. So because we can execute our simulations really fast within no time. So there are that were involved. This is my lab, and like there were several people that were involved when we did this. We formed a consortium in doing these, so I just wanted to thank TACC, BOINC TACC, and NSF funding for me. And other people who are involved with this. And Ritu, over to you.
Great. Well, thank you very much. Ritu, are you? I see you muted yourself, and thankfully, but now you can't unmute yourself. So give me a second. Okay.
Thank you. And I would like to take the opportunity to thank the community for supporting BOINC TACC, and also our colleague, David Anderson, other folks in the community like Marius Milia, on whose efforts we built BOINC TACC, and of course, TACC and Exceed, who provided resources to us for carrying out this project, and also the National Science Foundation that funded BOINC TACC project with David's help. And we look forward to transitioning from BOINC TACC to BOINC UTSA at some point in the future. So BOINC TACC project, its funding is set to expire on April 30, 2021. And then the next phase of the project, we are in the discussions to see how it can be sustained at TACC. And if possible, either we can move BOINC TACC to UTSA, or we'll be starting BOINC UTSA, a new project. So we'll be sending out information to the community and would be requesting your help to join us at BOINC UTSA once it's up and running. Thank you very much.
Great. Thank you. Thank you very much to both of you. So we've got a bit of time for questions. If anyone has any, please use the reactions and raise your hand, and we can see how that goes. Otherwise, I'll ask a quick question. I'm curious. You mentioned in your slides that as a preliminary, you needed to know the structure of the proteins of the virus and the binding site, and I'm curious where that information comes from.
Suman, that's a question for you.
I'm not sure if he's frozen, unfortunately.
Suman,
I think we might have lost him. He might have been disconnected, as happened earlier.
Maybe he needs to be unmuted. Maybe he muted while I was speaking? No?
I don't think so. Oh, maybe. Yeah, during these Q&As, by the way, please don't mute yourself as the speaker while the other person is talking, because the settings don't allow you to toggle back and forth. It's a one-way street.
Okay. Yes. Sorry, Matt. My connection was unstable and I lost you there. So can you repeat your question, please?
In your slides, you mentioned that as a preliminary to starting the work that you did, you needed to have knowledge of the structure of the proteins and also the binding site that's relevant to what you're doing. And I'm wondering where that came from. I think we might have lost him again. Okay, well, we have another question. Perhaps it's for you, Ritu. Let's see.
Maybe Suman is back.
Okay, I'm back. Sorry about this, but I think I got your question. So your question is about like, okay, that we need information. I mentioned that we need information about like, okay, the structure and the binding pocket. So yes, so well, of course, thankfully there were a lot of people working on this project or on this research. So we were leveraging a lot of published work. So there are structural biologists who are working on finding the structure of these viral proteins. So we are taking that information. So that is a lot of biology and structural biology work that was done before, and like we are taking the literature work and we are moving that forward. So the binding information, for example, we know like, okay, these what is the mechanism of these proteins? So like, what is their function? So like when we know the structure, now we know the structure and function. So we're trying to target the molecules that can fit in the structure and also block the function.
Great, thank you. So next up, we have a question from Mr. HH.
Okay, thanks for giving me the word. So the question I have is, docking is still less compute intensive than MD. So how do you do the communication and the computational resource handling if you do an MD simulation on a distributed cluster?
So is the question like how we run? So is that for Suman, right?
Yeah, so I can answer it, and Ritu, you may continue after that if you want to add some more. So for docking, yes, docking is less compute intensive, but so when we are one of the problem when we're running docking calculations on TACC clusters is IO problem. So and docking is smaller calculations, so we can actually split them nicely, smaller jobs, and send it through BOINC and TACC. So we were using both, but so these can be nicely split because we have billion compounds. We are splitting them into smaller chunks and sending it to the different computers.
But with MD, you cannot do that because you have causality. So how do you do it then? Because you basically have a time which you need to address.
Yeah. So, well, we did not run the MD on BOINC attack, but maybe Ruth can answer. But like it's so for the MD, even because like I don't know, Ritu, do you want to answer this? How we MD calculations were run on the BOINC attack?
So we did not run like for COVID-19 drug discovery. Every MD application for that one was run on Stampede 2 system. Okay. I think, but like for BOINC attack, we do support GROMACS, LAMPS, and NAMD. So far we have seen zero usage for NAMD, I guess, and zero usage for LAMPS. And for GROMACS, I think at some point somebody did run some tests, as in some researcher was wanting to see, and they ran it in single-threaded mode. And maybe that was a test job. It wasn't computation intensive or maybe memory intensive and IO intensive. So a majority of the jobs have been related to earthquake engineering, OpenSea so far, and Suman's work, which is like Autodoc Vina. Rest of the applications have been sporadic.
Okay. Okay, thank you.
My pleasure.